Concept 27.2 says rapid reproduction, mutation, and genetic recombination promote genetic diversity in prokaryotes. You're going to find that in these prokaryotic creatures, in these bacteria, in this archaea, there is an amazing amount of diversity. So we have to talk about how did that diversity come about. So we already know that for natural selection to occur, you have to have some genetic variation. That's a prerequisite. And prokaryotes certainly have considerable genetic variation. But you might want to ask, well, how do they get this genetic variation if they reproduce asexually? If all they do is divide in half and create two cloned daughter cells, where does this genetic variation come from? So we want to discuss that just a little bit. So when I say prokaryotes have considerable genetic variation, you might be surprised because we're talking about single-celled prokaryotic creatures. You might think they're pretty much all the same, but a good example is a ribosomal RNA gene can differ more between two strains of E. coli bacteria than it does between a human and a platypus. So there can be more differences in this one particular case we're looking at here between two types of E. coli bacteria than there are differences between a human and a platypus when it comes to ribosomal RNA. So that is the amazing amount of diversity that you're going to find within these two domains. Even though to us they might seem like, gosh, they're all alike, they're all just microscopic single-celled creatures, but they're not. There's quite a few genetic differences. There's actually three different factors that help contribute to this genetic diversity, even though, like I mentioned, for the most part they are reproducing asexually. One of them, of course, is rapid reproduction. We'll talk about how that affects it. The second one we already know, mutation. That's the original, basically the original fountain for genetic diversity is these mutations. And then genetic recombination. Although they don't undergo sexual reproduction, they don't have a mixing of genetics the way that we do in creatures that sexually reproduce. They do have something called genetic recombination, which is, I guess, as close as you're going to get to the type of sexual reproduction that we have in some eukaryotic creatures. So we'll mention that as well. So first let's talk about their rapid reproduction rates and mutation and how that leads to genetic diversity. We already know that prokaryotes reproduce very quickly. They reproduce by binary fission, basically dividing into two, creating two clone copy daughter cells, and the offspring are generally identical. Prokaryotes don't have the advantage of mixing genetics through meiosis and sexual reproduction and fertilization. So how do they get this genetic diversity that we're talking about? Well, we already know that mutations occur in all living things, and mutation rates are actually pretty low in all creatures, whether it's prokaryotes or eukaryotic creatures. But when the mutation rates, even though they are low, because these prokaryotic creatures reproduce so quickly and have such a rapid turnover of generations, these mutations, which don't happen very often, but if you have enough generations, they tend to build up very quickly. They accumulate rapidly in the population. So a lot of the diversity that becomes genetically available in the prokaryotes is from mutations. And it builds up quickly because of their rapid reproduction. And so that allows for a lot of mutations to accumulate, and that allows for rapid evolution. The second way that we can get some genetic diversity is through what we call genetic recombination. And although we have evidence that basically suggests that this process occurs both in bacteria and archaea, most of our knowledge about genetic recombination occurs in the study of bacteria, so that's kind of where we'll focus. Mutations are a major source of variation, but genetic recombination, which is the combining of DNA from two sources, also contributes to diversity. So in bacteria and archaea, genetic recombination is going to be, I guess, as close to sexual reproduction as you're going to get. So this is kind of a way of combining DNA from two different cells or two different creatures, but they don't really reproduce the way that, say, a cat or a dog or humans do. Prokaryotic DNA from different individuals can be joined together by basically three different ways: transformation, transduction, and conjugation. The first two we're going to look at, transformation and transduction, are really sort of accidental processes, and conjugation is really the only one that is like a purposeful sort of mating type procedure. There's also something we need to look at called horizontal gene transfer. That's the movement of genes among individuals from different species, and we're going to discuss that, how that happens as well. So genes can be transferred from one species to another in these prokaryotic worlds, and that's actually pretty common. Makes it more difficult to figure out which species is related to which other one and what are the phylogenetic trees, but we'll discuss this horizontal gene transfer in just a bit more detail coming up. So the first two ways that DNA can be combined between, say, two different creatures, two different cells, are sort of accidental ways. They're called transformation or transduction. In transformation, a prokaryotic cell can actually pick up and incorporate foreign DNA from its environment. So there may be bits of DNA or DNA from other creatures, DNA from other prokaryotic cells within the environment in which it lives, and that DNA can actually be brought into the cell, picked up, and then combined with its own DNA, and that's called transformation. That forms what we call a recombinant. A recombinant is a cell with a chromosome containing DNA from two different cells. So you could consider yourself, I guess, a recombinant because every one of your cells contains chromosomes from DNA from two different cells, from your mom and your dad. Well, when we're talking about prokaryotes, we don't have sexual reproduction, but if DNA is picked up from the surrounding environment and brought into the cell and combined with the DNA that's already there, we call that transformation, and then that cell is considered a recombinant because it has DNA from two different sources. The second sort of accidental way that this occurs is transduction, and transduction actually happens when viruses attack certain bacteria. There's a type of virus called a bacteriophage that preys on and infects bacteria, and when these viruses attack the bacteria, they tend to remove and pick up certain pieces of DNA and then infect another bacteria, sometimes putting that DNA into the second bacteria. It's very similar to the way that a disease bacteria or a disease material is carried, like for malaria or West Nile virus, transmitted from one sick person to another person by like a mosquito. So if you imagine on a smaller scale that the mosquito is the virus and it's picking up and transporting DNA from one bacteria cell to another, well, that's another way that DNA can be mixed within these bacteria, and that's called transduction. Your book gives you a nice little illustration. So here you have a donor bacteria cell and you have the virus, the phage, which infects the cell with its own DNA. That goes about transforming the interior to make up more viruses, so it breaks apart the DNA of the bacteria, and then you have more viruses that get created. One of those may contain a piece of the DNA from the original cell. When it goes on to infect a second cell, some of that DNA may be inputted into the new cell, and then that may become part of its own DNA, and you'll have a recombinant. So you see the DNA now is made up of not only the original but a portion that came from this donor cell. So we call that a recombinant, and this occurs not just in bacteria, it occurs in other creatures too. As a matter of fact, most studies show that part of our human DNA was brought to us via viruses, and so that may have happened earlier in our lineage. But some of our own DNA is viral DNA. It's part of this whole process that we see here called transduction. So those are two different sort of accidental ways in which DNA can be combined in a prokaryotic cell. Transformation from the environment, or transduction via basically infection by a virus. The last way in which we can increase genetic diversity in a prokaryotic creature is through conjugation and the use of plasmids. So this one is probably the only one that I would kind of consider as sort of purposeful, probably most similar to what happens in sexual reproduction. Conjugation is a process where genetic material is transferred between prokaryotic cells, and again, it's most like the purposeful mating that we see in eukaryotic creatures. Most of what we know about conjugation and this process has occurred from our extensive study of the E. coli bacteria. So again, although we think this also occurs the same way in archaea, we're most focused on the bacteria in this case. In bacteria, the DNA transfer is a one-way street. They're not really swapping DNA, but one bacteria cell is giving or providing DNA to another. We call a donor cell We call it a donor cell, which is one giving the DNA, and a recipient cell, which is the one receiving. A donor cell attaches to the recipient by a pilus. Remember, a pilus is that long, thin kind of a thread that connects the two, and then it pulls it closer, kind of like a fishing line, kind of like reeling in a fish, and then it transfers DNA. There's a particular type of piece of DNA called the F factor that is required for the production of the pili. The pili are these rods that we're talking about that connect the two, and you have to have the genetics to do that, to create that. And that little piece of DNA, that genetics, is called the F factor. F stands for fertility in this case. So here you have bacterial conjunction going on. This is the sex pilus that you see. This is basically the connecting point at which DNA will be transferred from one bacteria cell to the next. And sometimes that F factor is contained in its own little loop of genetic material, in its own little plasmid. So cells containing the F plasmid function as the DNA donor, and cells that don't have the F plasmid are the receiver, the recipient during conjunction. And the F factor itself, the little plasmid of DNA called the F factor, is sometimes what is transferred. It is transferable during conjugation. So this F factor consists usually of about 25 genes, and most of those genes are the genes that are required for the production of the pili to create this bridge between the two different bacterial cells. So here's a good illustration. At the top we have the donor cell, which is called F positive because it has the F plasmid. At the bottom is the F minus cell, the recipient cell. Here is the pili, the mating bridge between the two. The first thing that's going to happen is the F plasmid is replicated, and then one of those copies is going to be sent down the mating bridge to the recipient cell. Then you're going to end up with basically two cells that both have the F factor, that are both F positive, and they both have that plasmid. And now either one of these cells can go on to repeat that process with a F negative cell, a recipient cell that doesn't have the F factor. Sometimes the F factor is built into its own little plasmid, its separate little chromosome, little band of chromosome. Sometimes it is within the large circular chromosome that makes up the genome of the bacteria. So sometimes the F factor is not its own separate plasmid, it's part of the regular chromosome. And in this case, the cell that has the F factor, we're still going to call it the donor, and the one without the F factor is still going to be the recipient. But the recipient now is going to become a recombinant, because its main genome is going to be made up of a combination of DNA from two different creatures, itself and the donor that gave that piece of DNA. So the process works very similarly, except the F factor is going to be a portion of the major chromosome, the circular chromosome. That's going to be duplicated, and that piece is going to be sent to the recipient, and then that portion will become part of the main chromosome of the bacteria. So it will become what we call a recombinant. And here are the two kind of side by side. So basically these are ways, conjugation is a way of combining genetic information from two different cells, from two different bacteria. There are other plasmids beside this F factor, this fertility factor plasmid. For example, there's one called an R plasmid, and the R stands for resistance. And this was first studied and seen in the 1950s in Japan, where scientists noted that some patients who had a particular bacterial infection that caused dysentery, some of them responded very well to antibiotics, and it wiped out their disease no problem. Others did not really respond, and it turned out the reason was because the bacteria that these people were infected with had a resistance, an antibiotic resistance. So certain strains of the bacteria called Shigella had developed this antibiotic resistance. And what they had was this R plasmid. This plasmid carries genes for antibiotic resistance. And so antibiotics will kill off the weaker bacteria that don't have the plasmid, but may leave the stronger ones that do have the R plasmid. And that's basically natural selection at work. So the stronger ones survive and go on to reproduce, and you end up with a problem, because now the only ones in existence are going to be the ones that are antibiotic resistant. And we have seen this over and over again in the medical field, where different diseases, different bacterias, and other microorganisms have become resistant to the medications that used to be very effective. One of the main problems is that in many cases this same plasmid, this same little loop of genes that allows for antibiotic resistance also allows for that pili to be built to transfer this R plasmid to a different bacteria. So these heavy-duty antibiotic resistance bacteria can basically transfer that ability to another different bacteria that didn't have that ability, and that is going to be encouraged by natural selection. So that's a problem, and again, we have seen that over and over, especially in the last 50 years, where antibiotics have become normal routine procedures in the medical field. We've seen this resistance get worse and worse over time. In that case, we have evolution and natural selection working against us. It certainly is working in the positive for the bacteria, but it's not working out so well for the humans that the bacteria infect. We're going to take a break right there. When we come back, we'll get to concept 27.3. So take a quick break. When we come back, we'll talk about some of the other variety that we see as far as the nutritional and metabolic adaptations. And again, you're going to see a wide variety in these prokaryotic creatures. Take a break. Be right back.